So before we get started on movement across membranes, go into the content section of D2L, into the Module 3 cell module, which you can find right here. Remember, there's the resources folder and the to-do folder. So what I want you to do is open up this summary table, and then you're going to use that to fill in as I go through this lecture. And at the end of it, you will have a nice summary of the different types of movement across membranes.
So I took the table and I broke it into two halves so that we can kind of work on that here together. So let's talk first of all about these four types of molecular movement. So we have diffusion, osmosis, filtration, and facilitated diffusion. The first thing we need to determine is whether it's active or passive. Well, remember, active or passive means going to happen on its own, or we need to input some energy into it. So for diffusion, osmosis, filtration, and facilitated diffusion, these are all passive types of movement. So if they're passive, what type of energy do we need? We don't need any. For diffusion, it's really going to be anything that's small enough. So for example, if I burn a candle on the other side of the room, if somebody walks in the door right by where the candle is, they're going to smell those molecules right away. They're going to smell whatever the odor is. But it might take a few minutes for those molecules to diffuse their way over to me so that I can smell them. So diffusion is what makes Thanksgiving so amazing, because the whole house smells like Thanksgiving. Or for me personally, on weekends, if my husband wakes up before me and he's got bacon already in the oven for breakfast, the whole house starts to smell like bacon and coffee. Which, I mean, really, is there anything better? I mean, honestly. So that's all diffusion. Another example, diffusion is why you shut the bathroom door so that you don't share the diffusion of those molecules. Now with diffusion, the direction of movement is always going to be from high concentration to low concentration, or like you're sliding downhill. Osmosis is specifically diffusion of water. So now we're only moving water, and it has to go across a membrane. It's passive, so we're still moving from high to low concentration. Filtration is also a type of diffusion, but now we're talking about the diffusion of solutes. So now these are solutes, again going across a membrane, but we're still going from high to low concentration. Facilitated diffusion has diffusion right there in the name. So we know it's diffusion, so we know it's high to low concentration. But we're going to be doing things that are maybe a little bit bigger, like glucose, or some other kind of polar molecule that doesn't want to go across the membrane, like ions. So even though ions can be very small, like a sodium ion is only one atom, they don't want to go across that membrane because they're charged, which means they like water, and water's a little bit charged, right? That's why they like each other. So to get across that membrane, I must go through a protein channel. So facilitated diffusion requires a protein channel. All right, so everything on this table is passive, no energy needed, and we're always going to be going from high to low concentration. Let's look at the other types of movement. On the rest of that worksheet, we now have active transport using a transport protein, endocytosis, specifically pinocytosis, endocytosis, specifically phagocytosis, receptor-mediated endocytosis, and exocytosis. Are these active or passive? Well, every single one of these, I think we can agree, are active. So if it's active, that means we require energy. What is always going to be our go-to form of energy? No matter what, the thing we're using right now are those Duracell batteries. So ATP. If I'm using a transport protein to do active transport, these are typically going to be small things. Oftentimes we're transporting ions. Now it's active transport. So that means I'm having to push something uphill against its concentration gradient. So remember, a gradient is the difference of how many molecules you have from over here compared to how many you have over here. The steeper the difference, the steeper that gradient, then the faster you're going to ski down that hill. The closer the difference, so there's not as much difference between these two hills, then it's like going down the bunny hill, so it's going to be more gradual. But if there's a big difference, then I'm going to go down very quickly. In this case, I am trying to go up that hill. So that's what's going to require energy, or ATP. Endocytosis, and there are 3 different types, are when I'm trying to bring a whole bunch of stuff in all at once, or maybe I'm bringing like something really big in. So I'm going to bring it in through like a big kind of membrane bubble, because membranes are very fluid. Panocytosis means I'm bringing water in. And again, we said the direction of movement for endocytosis is always into the cell. Phagocytosis means cell eating. Panocytosis means cell drinking. So if I'm eating, I'm bringing in large particles or a lot of particles all at once. It's endocytosis, so again, into the cell. If I'm doing receptor-mediated endocytosis, then that means I have a whole bunch of receptors on my membrane. So think about the surface of my cell, and I have a bunch of mailboxes. Whenever you see this word, think a mailbox on the outside of a cell. So now I have receptors or mailboxes, and once I fill all those up, then I'm going to kind of pinch in the cell and form a bubble around those so I can remove these molecules. So this is when I have to have protein receptors on the surface of the cell, those mailboxes, and I want to bring in a lot of particles all at once. Typically things that are in low concentration outside of the cell. So I really want to grab whatever I can out there. Again, it's endocytosis, so that always means into the cell. Exocytosis then is the opposite of endo, so this means I'm moving stuff out of the cell, and these would be large particles or a lot of particles at once. So for example, if I'm a white blood cell, I might engulf a bacteria, phagocytosis, digest it using a lysosome, and then fuse that membrane bubble back with the outer membrane and dump the contents outside the cell. And that would be exocytosis. So review these different types of movement across membranes, and then what I want you to do is go into Connect and do 2 virtual labs. One is on diffusion and one is on tonicity.